Hello, my name is Osterholz. I'm a soil scientist with USDA ARS Soil Drainage Research Unit in Columbus, Ohio. I'll be introducing an ongoing research effort focused on connecting soil health to edge of field water quality in Ohio. First off, I want to be sure to acknowledge the contributions of Emily Duncan and Kevin King, who were responsible for collecting the data we're using in this project. A quick overview of what I'll discuss today. First, I'll introduce the water quality issues and discuss potential links between soil health and water quality in Ohio. And then I'll introduce the methods we used to measure both soil health and water quality in Ohio. And finally, I'll briefly explore some of the expected findings we have for this work. I won't be sharing any results here as we are still in the analysis phase of this work. And I'd like to start out spending just a minute to share the motivation we have for undertaking this work in Ohio. In Ohio, the conversation around the water quality impacts of agriculture is really centered around Lake Erie. Each summer, there is a large bloom of harmful algae in the lake. The primary driver of this bloom is phosphorus loss from croplands, primarily in the western Lake Erie Basin. So this issue has resulted in the state setting a specific target of a 40% reduction in total P and dissolved reactive P loads by 2025. In addition to the phosphorus issue in the Lake Erie Basin, much of Ohio falls within the Mississippi River drainage area. And so it contributes to the Gulf of Mexico hypoxic zone. This issue is primarily driven by nitrate. And while the state of Ohio hasn't set any specific nitrogen loss targets similar to those for phosphorus, the hypoxia task force has set a goal, a general nationwide goal of a 20% reduction in total end load entering the Gulf of Mexico by 2025. So in Ohio, there are issues with both losses of nitrogen and phosphorus, and we have goals for trying to reduce these losses. But in order to start addressing this issue, we need to think about what factors are important for nutrient losses from agriculture. So there are some inherent properties of the agricultural system that are going to be difficult to manage. Things like the hydrology and climate and the slope of an area. On top of that, there are management factors, for example, how fertilizer is managed, or certain attributes of the cropping system, such as the crop rotation or use of cover crops or no-till. In addition, the depth and spacing of tile drainage could be included in that category. Then finally, there are the soil-based factors. So these are things like soil infiltration, hydraulic conductivity, and the presence of preferential flow paths. So those are going to obviously influence how quickly water enters and moves through the soil and out of the system. There are also factors like the soil nutrient pool size or the stratification of those nutrients, which is especially important for phosphorus losses. And also nutrient cycling within the system, which can be both biotic and abiotic cycling. So these are the soil factors that are going to be important for nutrient transport. Now, there are a number of soil attributes that we can expect to influence these different soil factors. And each of these attributes is going to be connected, and they relate to one another in different ways. But I'd like to just discuss these 6 briefly. So soil organic matter is the one factor that probably has the largest overall influence, as it can impact both water movement through the soil, as well as it is a significant pool of organic nitrogen and phosphorus. It's also the base of the soil food web, and so it's going to drive the biological activity in the system. Soil pH can be important for determining what form nutrients are in. For example, pH is going to influence the interactions between soil phosphorus and soil minerals. Earthworms are going to influence water infiltration and movement through their burrowing activities. They're also going to drive internal nutrient cycling. Aggregate stability is going to influence infiltration and water movement, both through the soil as well as overland flow of water. And then water storage is obviously going to impact water movement. But in addition to that, soil moisture levels are important for determining microbial activities. And then finally, microbial activity itself is important to nutrient transport through processes like mineralization and mobilization that impact the forms of nutrients in the system. So these are sort of the attributes that we should be thinking about when we're trying to understand the connections between soil health and water quality at the edge of the fields. So I'd like to move into now discussing the particular study that will be included as part of the Journal of Environmental Quality's special issue. So for this work, we are working within the USDA ARS Edge of Field network. And so this is a network within Ohio that Kevin King has developed over the last decade, which is aimed at trying to understand the nitrogen and phosphorus losses from croplands in Ohio at the field scale. So you can see from the map here that there are 40 sites across the state, mostly in the northwest quadrant of Ohio. These were established between 2012 and 2017. And there are a whole suite of different agricultural practices studied as part of this Edge of Field network, including fertilizer management, cover crops, crop rotation, application of gypsum, drainage water management. So this is the Edge of Field network we're working within. And if you aren't familiar with this landscape of Ohio, it tends to be quite level, and the soils are mostly medium textured to fine textured, depending on the specific area of Ohio. But regardless, they tend to be poorly drained, and excess water is a major constraint to both timely fieldwork as well as crop production in this area. For this reason, the landscape has a lot of drainage infrastructure, and tile drainage is a very important part of the hydrology of the region. As you can see from this map, that northwestern quadrant of Ohio is one of the most heavily drained regions of the country. Okay, so now let's get into the methods a little bit. In terms of measuring water quality, each of these 40 fields is equipped with automated flow meters as well as automated water samplers. And for this study, we were focused on the time period of the water years 2017 and 2018. And so the data that we collect is processed into event-based nutrient losses, as well as flow-weighted mean concentrations. And we're looking at a number of different water quality constituents, including total nitrogen, nitrate, total phosphorus, and dissolved reactive phosphorus. In addition, we're also classifying the events by how large the flow for each event was. So we're splitting the data into 4 quadrants. So we have the lowest 25% of flows, and then the middle 25%, and then the highest 25% of flows. And we're doing this in order to see if the analysis results differ based on the size of the runoff event. Now, in terms of the soils data collection, there was a big soil sampling campaign in the fall of 2017 across the whole network. Samples were taken using a hydraulic probe, as you can see in this picture. And for this study, we were focusing on the top 15 centimeters of soil, and each sample was a composite of 5 individual cores taken within 2 meters of a central point. Each of the fields had multiple samples, and on average, there was about 1 sample per 2 hectares of field area. And then we simply averaged the sample values to calculate a field average value. Now, there were a number of measurements performed on these samples, including indicators of biological, chemical, and physical soil health. So biological indicators are shown in the blue boxes. So those were microbial biomass, which was measured using chloroform fumigation. Then also potential nitrogen mineralization, which was measured using aerobic incubation. Then in terms of the chemical indicators, that included the total organic carbon and nitrogen, soil pH, water extractable carbon, then H3A extractable nitrate, ammonium, and phosphate. And H3A is an extractant that's part of the Haney soil test. Then we also have Malic-3 extractable phosphorus, potassium, aluminum, and iron. And then in terms of the physical indicators, we have measurement of bulk density as well as dry aggregate stability. So I just want to quickly share the analyses we're currently working on. So we're looking at just the simple correlations between the various soil health indicators, as well as the correlations between those indicators and the water quality outcomes. And then in addition to that, we're constructing multiple linear regression models using a stepwise model building approach. And once we have those models built, we'll be looking at the partial correlation coefficients to try to understand how those soil health indicators were related to water quality. So the expected results of this work will hopefully include a better assessment of how soil health indicators varied across the edge of field network. Also, we'll have an exploration of how the various soil health indicators were related to the edge of field nitrogen and phosphorus losses and concentrations. And then hopefully we'll be able to answer whether the soil health water quality relationships differed under the varying flow regimes. So I'd like to take a moment to acknowledge all the contributors to this work. So the cooperating farmers, of course, are vital to the collection of this data. All of these edge of field sites are on working farms, and we have an excellent network of collaborators. In addition, we have a large team with the Soil Drainage Research Unit that makes the collection of all this edge of field data possible. And then we are fortunate to have a large number of organizations that have helped fund this work over the years. So with that, these are the references I cited, and thanks for your attention.